Guys, now that you have taken an overview of the cycle, that is the TCA cycle, now let's dig a little bit more deeply about different reactions of TCA. Before we see different reactions of TCA, let's see a little bit of the history of it. I mean, do you know what is another name of TCA? Yes, you are right. It's the Krebs cycle. Why? Because it was discovered in 1935 by Sir Hans Krebs. And the paper, I would say epoch-making paper, which was published in the same year, it really revolutionized our understanding of the biochemistry of life. The paper, as I have already mentioned you, that for a good student, especially for a good research student, and you all are research students also, you are not only doing your BS or MS to just take degree, you are also going to do research. So it's always good that you know about the history of any sort of discovery you are learning about, and also how and what sort of papers were written about that discovery. So this paper, which was written by Sir Hans Krebs, the metabolism of ketonic acids in animal tissues. It was an epoch-making paper, and because of this, in 1953, actually, the Nobel Prize was given to Sir Hans Krebs. Now, we formally start the very first reactions of the TCA. You know that in the glycolysis, the end product was pyruvate. Now, there are two fates that glycolysis can undergo. One is the aerobic and another is anaerobic. The next part of the aerobic glycolysis is to enter into the TCA, tricarboxylic cycle. For this, the pyruvate has to be converted into acetyl-CoA, a 2-carbon acetate molecule. So here the pyruvate, as you can see here, the pyruvate, which is a 3-carbon compound, it is converted into acetyl-CoA, and here the enzyme which is involved in this formation is the pyruvate dehydrogenase, a multi-enzyme complex it is. We will learn a little bit more about it very soon. Also keep in mind that in this reaction, NAD is converted into NADH2. In other words, you get NADH2, which will then go into the TCA, and you will get more and more ATP. In addition to this, it also have TPP, thiamine pyrophosphate, as the subunit, or as the coenzyme which help it to perform its function. Also, the energy you get out of this reaction is minus 33.4 kilojoule per mole. It's it's really very energetic reaction. This such a big low value or negative value of delta G, as you remember, if the delta G value is very negative, the reaction will be very spontaneous. Now let us learn a little bit more about ये pyruvate dehydrogenase complex होती क्या है. It is consisted of 3 subunits: pyruvate carboxylase 1, or pyruvate carboxylase rather, E1, dihydrolipoyl triacetylase E2, and dihydrolipoyl dehydrogenase E3. Each enzyme part, each subunit help in the overall performance of it contributes towards the overall performance of the enzyme naturally. Then, in addition to these three subunits, then there are 2 tightly bound regulatory enzymes which are attached with pyruvate dehydrogenase complex, and they regulate this complex. They are pyruvate dehydrogenase kinase enzyme and pyruvate dehydrogenase phosphatase enzyme. Both of these regulate the PDH or pyruvate dehydrogenase compound enzyme. Then there are certain coenzymes also. You have learned when we talk about the classification of enzymes that the coenzymes also help and contributes towards the functioning of the enzymes. Then this enzyme also have coenzymes like E1, the E1 subunits. It requires thymine pyrophosphate. E2 requires lipoic acid coenzyme. E3 requires flavin adenine dinucleotide and nicotine amide dinucleotide or NAD. So these are the different coenzymes which really contributes towards the functioning of the actual enzyme, that is PDH. Like any enzyme, its regulation is of absolute importance. It's not only that how enzyme perform its function, it's also important to learn how its functioning is regulated according to the environment in which it's exhibiting its function. So the PDH and that is phosphatase and kinases are the two enzymes out of which the kinase phosphorylate this enzyme and it deactivates, while the phosphatase dephosphorylate the enzyme and it activates. It's something very counterintuitive. Usually we talk about enzyme function in a way that if it gets phosphorylated, it will be active. Phosphorylation activity की तरफ लेके जाएगी और डिफॉस्फोरिलेशन इनहिबिशन करेगी, यहाँ उल्ट हो रहा है. So this is something very important to know, that phosphorylation may have different outcomes depending upon the environment in which the whole system is working. This is an allosteric activation or allosteric inhibition, of course. You know about allosteric activity of the enzymes. Then pyruvate dehydrogenase complex deficiency can occur, and it can occur, let's say, if any of the subunits, specifically if let's say E1 subunit have some problem or it's not it's not getting activated properly, so it will lead to problems. And one of the consequence of this is that you can face lactic acidosis. I mean, it must not be difficult for you to know okay, lactic acidosis क्यों होएगी, because the Krebs cycle is not performing its function, pyruvate is not getting into it's not getting converted into acetyl CoA. So what will happen? The pyruvate will be changed into lactate, and because the body needs energy, and somehow if one of the pathways is not working, it has to choose the another pathway. So the lactic acidosis will be resulted as a result. Also, arsenic, you hear on daily basis that arsenic in water or arsenic in food is poisonous, or it can be lethal, and we have to be careful. Why? Because arsenic usually used to inhibit the different enzymes or different metabolic pathways, and one of the most important target is the E2 complex of E2 subunit of PDH complex. As a result of this, the site which is available for interaction with the E2 is not really visible, or it's not available, and then the subunit cannot perform its function. For example, here the lipic acid is not available to serve as the coenzyme because of the arsenic binding with the subunit, E2 subunit. Finally, the citrate is formed. Once the acetyl CoA is formed, it reacts with the oxaloacetate. We will learn later that how oxaloacetate is formed, and as a result we get the citrate, and the citrate synthase is the enzyme which is used here. This reaction takes place because of the very high equilibrium in the direction of citrate synthesis. I mean, citrate synthesis is very spontaneous, and because of this, because of course the value of delta G is such that the reaction can occur very easily. In human, citrate synthesis is inhibited by its product. It's not only human, I mean, and generally through the feedback mechanism, the product can inhibit its reaction.